Good afternoon, everyone. Welcome to the first seminar of our series for the term. Our speaker today is Ercüment Çiçek of Bilkent University. Ercüment Çiçek got his MS and BS degrees in computer engineering from Sabancı University in 2007 and 2009. He got his PhD from Case Western Reserve University. After graduation, he was a Lane Fellow in Computational Biology at Carnegie Mellon University until 2015. Since then, he is an assistant professor at Computer Engineering Department at Bilkent University and an adjunct faculty member in Computational Biology Department in Carnegie Mellon University. He is the recipient of Simmons Foundation's Autism Research Initiatives Explorer Award and Pilot Award. He is the recipient of TÜBİTAK Career Award and TÜBİTAK Gelip Award and Parla Foundation's Research Incentive Award.
Thank you very much. Hello everyone, thank you very much for the invitation. Today I'm going to talk about our recent work, which is about selecting predictive and diverse SNPs in genome-wide association studies. This is the first time I'm giving this talk, and in particular to the EEE department, so I'm sure there are many unknown keywords here. So hopefully we'll figure that out. So if something is not clear, just stop me please. So this is recently published in this journal, and then this is the QR code to the paper if you're interested. It's going to be available throughout the slides. And this work is a joint work with Özgür Taştan, who is at Sabancı University, Computer Engineering Computer Science Department. And Serkan was my master's student. He's doing his PhD overseas right now. So first, let me explain what this GWAS, Genome-wide Association Study means. So if you have a condition, basically it might usually it's a disease, and you suspect it has some genetic ties, what researchers would do is to gather as much as people with this condition and sequence their DNA, and that would be the people with the condition here, and then gather some control people who are basically without the condition, gather their DNA, and then try to see which genetic alterations cause this disease. Because so it is not enough to get the DNA sequences of the people with the condition, because so in humans there are like 3 billion letters in the sequence of the human DNA, and then usually there are like on average 3 million mutations, which actually also is what the SNP means. It's single nucleotide polymorphism. So each and every one of us has like 3 million mutations, and then only a very small subset of this is going to be causing the disease. So what people do is it's not enough to get the DNAs of the people with the condition, because you don't know which mutations cause the disease. So you get a group of people with the condition and do this control study, obviously, and then try to see which genetic alterations cause the phenotype. And then usually what you generate is a plot like this. Has anybody seen such a plot or know the name? Alright, so this is called a Manhattan plot. Obviously it looks like the skyline of Manhattan. And what you see on the x-axis is the human chromosomes here, 20 up to 22 and the X chromosome. And then the positions here are the basically nucleotides on each chromosome. And then what you see over here is the mutations that you have observed in the cases. So they build up like buildings, right? And then there's like a significance threshold. You hope, given your study size, the control and the case size, the number of SNPs that has occurred in the cases, meaning the people with the condition, as opposed to controls, to reach up to and exceed this threshold. Then you say these mutations, so you see some names over here. These are the genes that has overwhelming number of mutations in the cases as opposed to controls. So these are genome-wide significance mutations, and then the genes are quite likely to be responsible for the condition, whatever the condition is. And then this is the GWAS catalog of the human studies. Basically, you see again the 23 chromosomes here. It is very small, but for every position on this chromosome, you see which conditions have been associated with which. So which SNPs are associated with which condition, which human disease, so far. So I mean, it's a frequently done study to study in particular human disease. You can also do this. So this is, in this case, your condition is discrete, binary, right? You have people who have the disease and people who do not. You can also do this for continuous traits, and this is going to be my example today. So you are interested in which mutations lead to this plant's flowering time, or for instance, flowering time. You want the plant, for instance, to produce flowers earlier. So which mutations cause this? So you can do some genetic engineering maybe to have better plants, right? So in that case, it would be continuous. Now, our goal in this study is to identify a SNP set, a mutation set. Given all the mutations in this study, I want to select a small subset. So on average, each individual has 3 million, I said, right? So you union them, you expect it to be around 3 million. I'm given 3 million mutations, and I'm looking for the causative ones that lead to this phenotype. So our problem is to select the smaller subset which is predictive of this phenotype. Phenotype meaning you have some genetic condition, and then you observe some change in the person, in the sample, just like the disease. So I want to find a small subset which is related to this phenotype. So there are some challenges associated. First, it's rare to have such an. If it is a monogenic disease, meaning a single gene causes the mutation, then you would have just a single peak over here, just a single building, and then because everybody in your population has the mutation in the same gene. It's an easy case. It's a monogenic disease. But not all genetic diseases are such easy cases. Just like, for instance, in autism, we expect 1,000 genes to be related to the disorder, and then everybody has a different combination of mutations that give you similar phenotype. Now, so then what you observe is, like nobody passing this threshold. You don't know which gene really causes the mutation. That's a problem. Another problem is we have low sample sizes. The studies are up to 10,000 samples usually, and it's very common to see a sample size around 100. And we have very high dimension. And then there are problems to select the subset and select the meaningful features that explain the phenotype in these challenges. Now, so what are the approaches to here if we don't have these standing out SNPs? So what you can do is just we wish for the best and try the pairs. So which two SNPs? If I don't have individual SNPs explaining the phenotype, then I might just look for groups, and then we are trying to select subsets. Let's focus on just the pairs, right? Still, just too many computations. This is called epistasis testing, and it's computationally challenging, so you cannot do it. The second approach would be so when I'm trying to select mutations among 3 million, so I'm trying to select a subset of 3 million mutations, then maybe we just focus on the parts of the genome which are active. By that I mean, why not we just focus on genes? So, the human genome, 99% of it is not coding, meaning only 1% produces proteins which directly affect function. So it used to be called the junk DNA, if you remember from old days. So 99% of the genome basically does nothing. It's not the case. It has regulatory function. But really, the 1% is the part which directly affects function by producing proteins, just like high school biology was. Okay. So what we can do, let's focus on the parts of the genome, the mutations that fall on the active parts of the genome. so group mutations that fall onto the genes, and then do your statistical test based on the genes. So group mutations, and then do the test to reduce the search space. But then still, first we have like 20,000 genes in our genome. That's one problem, and it also leads to literature bias, meaning you are over-constraining your search space only to the active parts, but you know the non-coding parts, inactive parts, really has ties to the condition. And, you know, I have a slide that shows that result as well. So then what can we do? Even here you can group genes, right? So some genes do the same function, so they are grouped, their pathway, that's what it's called. So you can even do your analysis based on those groups, but you know, again we have the literature bias. So the third approach, which we focus here, is that let's account for those interactions not on the basis of parts of the genome that interact in terms of active parts of the genome that interact, but do an analysis on a network of the mutations themselves. So don't constrain yourself to the coding images, but have a network of related mutations. So that's what we call the SNP network. And then, so we consider these, we construct these with respect to some a priori information, and do your search here. Hopefully it's going to give you a restricted search space, and it still is going to have some biological background, right? That's all. So the first and actually the only example of this approach is the SCONS method. It means selecting connected and explanatory SNPs. Okay, so it first it accounts for the genetic interactions using such a network. So we have a network here in which the circles are the SNPs mutations, and then somehow they are related. I'm going to show you what that relation means. And then the goal here, so this is an edited model. So every mutation has an individual relation, like a univariate relation to mutations, like a price for each mutation over here. And the goal of this algorithm is to select a connected set of SNPs. So it can be like multiple groups just like this, but the optimization algorithm enforces you for these mutations to be connected. So this falls back onto the guild by association paradigm, meaning if two things are interacting, they have something in common. That's a very common assumption in the biology domain. So if, for instance, you have a network of genes or SNPs, if you know one to be related to the condition, then the neighbors of that variable, whatever it is a gene, SNP, they are likely to be also related because of guild by association. Yes. So this is the idea. So if I know a selected gene, here it's a selected SNP mutation, I should be likely to select its neighbors because they are likely to be related as well. So this is advantageous because you can select SNPs from non-coding inactive regions, and maybe they are also related, you don't know. And this biological network connectivity constraint provides a biological interpretation. So you know the literature says you can use lasso, for instance, for this purpose, but then there is no biological interpretation of the coefficients of the variables. So each, in this case, every variable is a SNP. Just do lasso, you will have a sparsity, but then it's not informed with the biology that variable does. So we can do this. Then our issue with this is that the opportunity cost with the phenotype prediction, since we are trying to select the SNPs that best explain the phenotype, right? We are limited, so we limit ourselves to a small number of selections for interpretability, right? So I'm given like millions of mutations. I want a small subset which explains the phenotype best, and then maybe I'm going to focus on that subset for further biological interpretation. What do these mutations disrupt so that I have this condition? That's our now. So this is the optimization function of the SCONS. So it tries to find the f that maximizes this function. Here, so these p to n, these are all the mutations that we have. And let's focus on the left part. So CP is the price of every mutation. So it's basically a univariate test. You check how often you see this mutation in the cases as opposed to controls. This is a normalization parameter. So this is for sparsity. You basically subtract a constant from your price. Then it encourages sparsity. This FP is a binary indicator variable, so it is 1 if you select this mutation, 0 otherwise. And then this part is the penalty part, which penalizes if the two SNPs that you have selected, so rather, let's say you have selected P, and then you haven't selected Q, this is going to be 1, right? So this encourages you to select, if you selected one SNP, you have to, or you should prefer selecting its neighbor. Otherwise you are going to pay a penalty, right? So this is going to be 0 if you don't select them both, or if you both select those SNPs. So this is what they try to maximize. So this is NP complete, and then they formulate this as a minimum cut finding problem. So they can solve this with a maximum flow algorithm, which is based on Tarjan's 80s algorithms. But the bottom line is that they have an efficient solution to solve this problem. Now, so this is the literature. Again, to sum up, the goal in the literature is to find a connected set of mutations with respect to a biological interaction network, so that you have the biological background as well. So one problem with this approach is that depending on also how you define your network, two SNPs that are close on the genome might be related, but not because they are both contributing to this condition you have, but just because they are close on the genome. So what do I mean by that? So once we inherit DNA from our parents, you don't inherit each and every letter independent from each other. Rather, DNA are inherited in pieces. So if you have two close by mutations, and if your mother or father has that, and then if that block transferred to the child, those mutations are going to be transferred together. So both of them occur once you have the disease, but it's not like they have been selected independently and they have to be both there to cause the disease. You know, maybe just one of them is related, but the other one is just its neighbor, and then because you inherit chunks of DNA from your parents, it comes with it. So what's the problem here? The problem is, if I have a limited space of mutations to select, and if I select these two mutations, they carry redundant information. I only need one, and I waste one of my spaces for my neighbor, right? So, and I want as much as complementation. So maybe like multiple mutations lead to this disease. So if I have like 1,000 spots in my set, I want like a water from different causes. So if you prefer the scones approach, you enforce your algorithm to select close by mutations, and then if your closeness also means, you know, the genomic location closeness, then you might run into this problem. You get SNPs that are redundant, that cause the same effect altogether. So our idea with the SPADIS approach is, if you select SNPs from close by regions, then you have redundant information. Why don't I diversify my selection set? So what we do, if we select a mutation from this location, we basically in our formulation penalize the neighborhood. So if you select one, and each one again has an individual price, if you select one, all the neighborhood of that selection is going to be penalized, so that you are going to be less likely to select that mutation. Rather, we want to go far away, so to diversify our set. So this is like if you build like a base station for a cell phone, you don't want a second base station to serve the same area. Right? You just go build it somewhere else so that it's going to have an independent coverage area, very similar idea. So we basically do the opposite of what Scones does here. Yeah, so these are some networks that Scones algorithm uses. So the first one is just the genomic sequence clone. So each number here is a SNP, and then they are linked if they are adjacent on the genomic sequence. So this is their first approach. The second one is the GM network, in which They are connected if they are neighbors on the genomic sequence, plus they are near the same gene. So if this is like a coding region, an active part of the genome, so they might have a functional relationship, so you also have a clip for all the SNPs within this gene. The last one is you also link SNPs that fall into genes which are related, that you know function that they are related. Now you also link 9 and 2, even though they are quite far apart on the gene. So we also tried here a specific type of data in which we also consider the 3D conformation of the SNP. So this is a relatively new technology in which you can find out how the DNA is folded within your nucleus, right? And then it has been shown this as functional consequences. So if you have DNA closed and then the inner parts of the DNA is not going to be active, they are not going to be able to produce proteins out of there, right? So mRNA to protein, that's what I mean. And you have to open up the DNA at least that part for that part to be active. So people now measure, and they measure and then plot how the DNA is folded. So if, in addition to in one dimension, if in 3 dimensions 2 parts of the genome are quite close, then they have a functional relationship just like over here. So, so in this case, the 21 SNP is close to 2 and 3, so they have some functional. They are likely to have some functional impact. I'm not going to go into biological details, but this is the case. So this folding, these loops have they are called topological domains. So they have functional consequences if it's disrupted. So we also now have a fourth network, and we try to see if this helps with the selection process. Now, this 21, in addition to this network, now 2 SNPs are related if they are close, significantly close on 3-dimensional conformation. So we only use intrachromosomal contacts, so you can also have the contacts between chromosomes, but it's quite noisy, so we discarded that. And then we use the method called IEEE, which is over here to assess the significance of this contact. So is it by chance that they are close, or is it a really significance? And there's a significance there, meaning so you want the points that are, so if 2 points are close in 1 dimension, they are close in 3rd dimension as well, right, when you consider 3 dimensions. But you want the SNPs that are close in 3D, but not in 1D, so that you have those. Anyway, so we generated this. So this network says if 2 SNPs are adjacent sequentially, it means now they are in linkage disequilibrium. This is what it means to have inherit DNA in blocks, so that 2 SNPs are related to the phenotype, but one may by chance, only one of them is responsible. And then once they are close in 3D, it means they have some functional similarity. So this is the kind of our contribution to this network. And then this is the input of our algorithm space. So we have X matrix, which has the samples here, and then the features SNPs over here. It's a binary matrix, so if this individual has this mutation or not. In our test, we are using a plan, so it has only one copy for each chromosome, but in humans this can get complicated because we carry 2 copies of the same chromosome. But for now, we just simplified, just if you have that mutation or not, that's our point. So this is the genotype data. Then we also input a SNP by SNP network in which this is going to be 1 if 2 SNPs are linked with respect to the networks that I described, whichever network that you choose. And then we have the number of features and number of samples, and then we have, of course, this problem. So many more features than the number of samples we have. So how do we proceed? So we first input the X and Y matrices into, and then I forgot to mention Y. Y is the phenotype, basically, if that sample has this phenotype or not, or if it's continuous, it's also okay. So if you have the, for instance, the flowering time, so how much time it takes for this plant to flower, for instance. Now, the first thing we do is that we input this x and y into this box, in which we calculate univariate scores. So for every individual SNP, we measure if it is related or not. Okay, but then we are basically losing the big picture, their interactions, but assuming they are independent, how much each and every one of them are related to the condition that you have. So this produces a vector which is called C, individual scores for every SNP, and then in the second step of the algorithm, we are performing an optimization problem in which now we input the W matrix, which is basically the adjacency matrix of the mutations. Then here we compute a function, optimization function f, and trying to maximize it given a condition, given a cardinality constraint, meaning I don't want any more SNPs than k, like 1,000. We are trying to find a subset of size that's smaller than k, and such that it maximizes our optimization function f. So here the C rewards the phenotype association scores in f. I'll show you the formula. Basically, I want to select SNPs that have high price in terms of their individual linkage. And the second part, we are going to penalize the close proximity of the selected SNPs on the network that we use. So this is going to be important. Let me go back one step. The problem is that the subset selection problem is NP-hard, right? So we have a set of V vertices and then selecting a subset of size k. This is NP-hard. So we are trying to get away with some approximation. Our optimization function is sum modular, and then this lets us to use an agreed algorithm which guarantees us to have a solution, a constant factor approximation to the optimal solution. It's going to come in a second. So this is our optimization function f. Now the S is the selected set of SNPs. So the left part sums over the prices of the SNPs that we selected in the set so far. So we try to maximize this again. And then this beta is basically a parameter that adjusts the relative size of the SNP price compared to the penalty that we get for the neighborhood. So this is the part that we pay price for the selecting close SNPs. Here, so it's a nested summation, and then we keep summing over all the selected SNPs in S. And then this Kij is going to be the penalty function. So this is just for normalization. Due to symmetry, we have 2 over here. So because we count the penalty for Ij and Ji in this formulation, just for the ease of observation. That's why we divide by 2, and then we divide by the size of the selected set just to normalize. I'm going to show you what K is. So this is the definition of Kij. Now, 2 SNPs Ij are considered close if their shortest path distance is smaller than a second parameter d. So basically what I'm saying is I'm inputting a parameter, and then if I select this SNP, if the SNP is me, I have like a circle around me such that if I select this SNP that is k hops away from me, I'm going to pay a price for it. So if the distance of the j SNP to the I SNP is larger than d, I don't pay a price. It's going to be 0. If it is less than, the shortest path is less than my parameter d, then I pay a price which is proportional to the distance, basically. This is what it says. And then this beta parameter adjusts that number. And our goal is to have this set such that it maximizes this function. Questions? Comments? Yes?
We have many parameters. How do you set them?
So we have 2 parameters. So it's kind of be unlucky here. But I agree it's still a big problem. So firstly, the experimental setup is quite like it takes like a column of the paper right now. First we select D, assuming, it's quite detailed, we can discuss offline, but first we select D, and then with respect to the best performing D, we select beta. And then we are trying to play by the rules of the scones algorithm. So what they do is they have like 7 options for each parameter in their task. We do the same. First we try some values for D, and then we, with respect to the best performing D, we select among the 7 options for beta. Just to, but agree it's a promise.
How about the cardinality constraint, K?
So actually in the paper we also got that comment. We take it as an input, we don't optimize for that. The first reason is, actually there are many reasons. The first reason is we think that the geneticists who would use this algorithm would have an idea about how many SNPs they would ask, right? Given that, so they don't want any more than 1,000, for instance. So that's the first reason we take it as an input. The second one is that our optimization function is sum modular, so it's guaranteed to increase with decreasing speed. So if as you add more, it's going to increase. So still we have a point in taking it as an input. Plus, once we select the SNPs, we measure the performance of the algorithm using ridge regression. So that ridge regression can also still eliminate some of the SNPs that we have selected. So we do a further feature selection there. But you know, the main reason is that there are too many parameters. But we would love to optimize for it. Thank you. Any other questions? Okay. So, alright, so I told you that this F is sum modular. For me to be able to prove that, I think I still have time. So first let me show you the gain function. So sum modularity is basically the concavity in discrete verbs. It means, let me get to the sum modularity definition in the next slide, but let me first introduce you this gain function. So I give you an S, an item to be added to this, to my set. It is basically the difference between my function when X is added minus when my set is without X. So basically this part is same as this part with the exception that my S now includes X, right? So if I subtract this number from this number, this is what I get. So it is the individual price of X plus my beta minus the price that I pay for X for its closeness to all items in my set S. Right? So this is going to be important in a second. So a sum modular function F, so it's defined on sets, right? We have a limit, we have a fixed set of mutations to count from. My goal is to select a subset of it. So my F is going to be sum modular if the gain function on A is larger than or equal to gain function on B when I add X. So this is what I get, this is the gain function. For all sets A and B, such that A is a subset of B and B is a subset of B, this is the total set of SNPs. So what it means is, if I'm adding X onto a small set and add it to a superset of this small set, the difference should, the gain of adding that SNP or that variable should be higher for the smaller set. Right? So we have the diminishing returns property over here. As you add more items into the set, your gain is going to decrease. So that's the idea. And then here we prove it, it's not that important. This is the gain function for the small set. This is the gain function for the superset. And if you subtract this from this, this is what you are left with. So if you remember, this is the penalty I pay for X's closeness to I. And here I am left with all the members in B that A does not have. So I have the difference of B and A over here. And so this is a number between 0 and 1. Right? So this each and every one of them. At most it can be 1. I guarantee that. So K is larger than 1. Larger than or equal to 1. B is a parameter which is larger than or equal to 0. So I am guaranteed this is going to be larger than or equal to 0. So my gain for A is larger than my gain for B, right? So this proves that my F is sum modular, and if I also guarantee that this sum modular function is non-negative and it's monotone, then I'm going to be able to use a very simple algorithm which guarantees that approximate solution. So that's the gist of it, why we just try so hard for this function to be sum-modular. So this is my gain. So I have to prove you that my gain function is always larger than or equal to 0. So as I add more synapse into my set, I'm going to keep increasing the value of my function f. So this is the gain function. If you actually this is the same thing as this k over here. This can be at most 1, this k xi. Right? So I just input 1 over there, and then I sum for all the synapses in my S. So this is the cardinality of S. Then I'm left with 1 minus S over k. So I'm guaranteed the set that I select is going to be smaller than k. So this is smaller than or equal to k. And this guarantees that my gain function is larger than or equal to 0. And I know if I have an empty set, it is just 0. The function's value is 0. So as I add more synapses, I'm guaranteed it's going to stay the same or increase. So I have a sum-modular function f. I proved it's sum-modular. And I prove it's monotone and non-decreasing. Right? Then we have this nice algorithm from 70s. Very interesting, right? So it says if this function is defined on sets, if it is sum-modular set function, non-negative and monotonically non-decreasing, then basically if you use this simple greedy algorithm, which tells you put the synapse which maximizes your function at that point into the set, keep doing this. It's a very simple algorithm. If you do this, it tells you the set that this greedy algorithm returns, its value is going to be larger than or equal to your optimum solution times this constant factor. So we cannot find the optimum solution, but we are guaranteed to be close with a constant factor approximation. So that's why we try to do, stay within the bounds of some modularity. Questions? Yes.
What is the value of E in general? The closeness, I mean, to the optimal value. E determines that, but is it like a small value?
That's the E, natural one. The natural
Oh,
one.
that's
Yeah.
okay, not a constant.
Yeah. I mean, technically we didn't really check how close. We didn't search for the optimum solution on a very small set actually. But what we really cared about the phenotype prediction. This is just a guarantee that we have, like a cycle. In biology, usually we try to as much as we can, but people really focus on the performance rather than the balance. Any other questions? Alright, so let me show you some results. So we are dealing with this Arabidopsis thaliana. This is a type of a weed. Right? So we have close to 200 samples, close to 200,000 SNPs, and then we have 17 different continuous phenotypes here, all about the flowering time of this plant. And then we would like to, given this many features, we would like to select a small subset which gives us the best prediction in terms of predicting the flowering time. So we led the methods to select at most 1733 SNPs. This is like the 10% of all SNPs corresponds with the same too. And then here you see the average performances of the methods on 4 different network selections. And then this scones R, scones S, lasso S, these are different optimization. So how do we select the best parameter setting for this method? This version optimizes for the regression performance. That's what it tries to select the best parameters that optimizes the regression performance. This S optimizes for the stability. So in different folds, select the synapse that you select in all folds, kind of, right? So if you select the same set of synapses in different folds, it means those synapses are stable. So use those, something like this, right? So we tried different strategies over there. We have this univariate over here, basically just select the synapses which are individually most related and so on. So looks like the SPADIS is the top performance, giving the top performance in all, when you average over all phenotypes. Then we focused on scones, and scones was the second actually, and it makes sense because it's focused on this topic, right? So we checked for the improvement that SPADIS provides, which is the red part over here, whereas the blues are the scones result, given different K values. And then here, basically, you have different network types, and then this again average for all 17 phenotypes. As you see that our improvement is much larger when K is small because of the submodularity, right? Because our gain, even in our function, is much larger when we have a small set. And then as we keep adding, it kind of converges to what scones wise, but still, even at 1,000, selecting 1,000 SNPs, we are improving over scones. And then these are different details about experimental settings, but at different K, and for instance here we enforce scones to select 100 SNPs just like us, whereas in this version we let it select something between 1733. So different settings, so we check if our results are larger, I mean we improve significantly over scones using a two-tailed two-tailed t-test over here. And we find that in all experiments we are significantly better than scones. This is the same figure, but now to see if we really, the performance improvement we get is due to this diversification. So we checked the number of different genes our selected SNPs fall into. Right? We try to avoid selecting close SNPs, so we expect the number of genes that are hit to go up as opposed to scones, and then that's what we see over here. As we add more and more SNPs, you see that we select SNPs that affect more genes, and then we expect if you hit more genes, you select SNPs that are diverse because the genes are likely to have different function effects, right? So this kind of supports our idea that diversification is the reason in the increase in the prediction power. These are some other numbers. So this is the gene hits metric that I showed you, the number of genes that we have at given different K numbers as opposed to other methods. So this is the go hit. This is like an annotation for the genes. So for each gene, this go ontology assigns it a function. So now we are like one level up. We are trying to see how many functions we have hit as opposed to the other methods. So we are hitting much more functions compared to scones again. So this is a metric that was introduced by the scones method. So it's the within gene set. So how many of the mutations that I select falls into a gene and a coding region. That's what it checks. So it says if the SNPs that I select fall into a gene, I'm doing good. It's like something like a precision. How many of the SNPs that I selected are falling into a gene? Right? So we are performing really bad here, and scones in both settings are beating us. But if you think about it, if you have, say you selected 1,000 genes, you have selected 1,000 SNPs. Each and every one of them are falling into a single gene. Then you would have a within gene set metric of 1. But that's not our goal, right? So we want to diversify. We are performing poorly here, but this is not a good metric in terms of measuring if the method is really doing well in terms of predicting the phenotype, which is our goal. So let's check that. Okay, I still have some time. So we got a comment on saying from a reviewer which said, so you claim that SNPs that are falling into non-coding regions are also useful. So this within genes metric is not that good, right? So can you compare if the SNPs that fall into non-coding regions are also informative? So this is for different, so some subsets of the 17 phenotypes that we considered. These are the individual scores of the SNPs that we get. and then this is the distribution. So these are the negative SNPs, meaning falling into non-coding regions. These are the positive SNPs that fall into coding regions. And then you see that their relatedness scores individual are, these are actually white on top, but not look like a white right now. So this is basically a small jitter over here. It's just a random number to show you the effect. What we see over all phenotypes is that the non-coding SNPs are almost as related as the coding SNPs to the flowering phenotype here. So that which supports our claim that this within genes metric is not a good idea, and then you should also not restrict yourselves to the coding regions and then focus on to the non-coding regions as well. So this is a runtime performance comparison for one of the folds. Actually, it takes a lot of time, but we are better than scones and GraphLasso. These are even not feasible to run these algorithms. Lasso, yes, but not GraphLasso. This should be log scale from here. So we are worse than univariate, but this is a very simple. Basically does nothing, and then so we are performing best and performing better with smaller runtime requirements. Last thing that I want to tell you is that we checked if this 3D data has given us some performance advantage, and this is the average for both the scones and SPADIS algorithms on all networks and all, on all phenotypes, but we show different results for different networks. We see that this 3D information that we get improves performance on average, only not in this setting, but it actually helps scones more than us, because if you think about it, if we let scones do, so scones selects close by SNPs, right? So it has this locality constraint, it cannot get out of it. But if you add this 3D dimension, now the portions of the genome that are quite far away, they are now close. So it's allowed to select them as if they are close. So it improves scones performance more than us. Okay, I think I'm kind of out of time. So let me skip this summary and then thank my master student Serhan and also Tashan from Saran University. So thank you. I can take questions if you have any.
Ben devam edeyim. Aslında bu genel bir soru. Şimdi performansı correlation coefficient üzerinden gösteriyorsun. Başka metriklerde söyledin ama yani biyolojide çok kullanılır bu correlation coefficient'lar. Ben şeyi çok merak ediyorum. Hani correlation coefficient hangi değerin üzerindeyse siz buna evet burada istatistiksel olarak güvenilir bir ilişki var diyebiliyorsunuz ya da diyebiliyor musunuz?
Diyemiyoruz. Evet, maalesef yani yapabildiğimizin en iyisini yapmak gibi bir amaç var. Zaten yani çok kompleks bir problem olduğu için, yani milyonlarca veri bunun olduğu bir durumda neyin üstü iyidir diye hiç bakmadık herhalde. Evet, hiç onu consider bile etmedik. Sadece var olan metotların üzerine ne kadar improve edebiliyoruz ona baktık. Sonra farklı amaçlar için de kullandığı zamanım kalmadı aslında. Bir de bunu şey için de, hani pair'larını seçmek için de bu fikrin çok iyi olduğunu bulduk. Hakikaten de complementary region'ları seçiyor ama evet, ona bir cevabım yok maalesef.
Rakam olarak 0.4, 0.5'ler yani hani
evet.
Çok değil aslında hissi veriyor bana ama hani...
Çok değil tabii ki, çünkü bir şey de yani bu flowering time'ı etkileyen şey de var. Genetik olmayan koşullar da var, değil mi? Biz sadece yani ne kadar portion'un o sebeple oluştuğunu dahi bilmiyoruz. Bu yüzden yapabileceğimiz, yani genetik olduğunu düşündüğümüz kısım için en fazlasını predict edip işte genetikçilere bir subset sunabilmek.